observed, oh, good. We also observed that the anisotropy ratio is different than one. In this case, the anisotropy ratio is the ratio between the viscosity when measured at the direction orthogonal to the surface of the cell, and the viscosity measured parallel to the surface of the cell. No? And generally, we observed that there were deviations of what should be expected, again, according to the Faxen's law. Finally, we wanted to see what would happen if we induced lysis on cells. We have here, this is a video from a really nice paper from Smriga in 2016, showing, well, they used UV light to induce the lysis of, the lysis of a Chaetotheros cell, and then they could see how immediately when they released all their content, all the bacteria would swarm around the lysed cell. No? So our expectation was that as they release all these chemotactins, they are also releasing a lot of the cytoplasm, which is viscous and way more viscous than water in any way. So we replicated the same procedures than they, and this is the result. So this is just an example video or GIF showing these viscosity maps around a Chaetotheros cell that was lysed using this method, and they are separated about 20-25 minutes apart. So each one of these frames is 20-25 minutes apart, and you can see how, in general, there's this kind of lump of viscous material that is going out of the cell. Generally speaking, we observed that after lysing, the whole frame became more viscous. So it's probably that not by much, not around about twice more viscous than seawater. It seems that all the aggregated contents of all the cells that were lysed within that zone were increasing the viscosity of our sample. So the question is are these what we call them viscospheres common or not? And I have to say we don't know, because so far we have only worked with phytoplankton cultures, but it was remarkable how common they were within our universe of samples. No. So we tested Alexandrium minutum and what's it called, and Ostreopsis ovata, which are dinoflagellates. We tested it also in Chaetotheros affinis and Skeletonema pseudocostatum, sorry, and Cylindrotheca fusiformis, which are diatoms, and also on colonies of Phytoplanktons globosa. No? And this graph here tries to summarize all the results we got from this study, no? Showing the relative viscosity close to the surface of the cell against the volume of the cell. And that was something that was a little bit surprising to us, because we were expecting that there would be a correlation between the viscosity of the water surrounding the cell and the extent of this viscous phycosphere with the size of the cell, but we didn't find any correlation. We did detect viscosity, these viscospheres in almost all the species except for Cylindrotheca fusiformis. I'm not going to enter into details with this, but there, we suspect that there was a lot going on in terms of nutrient limitation and the physiological status of the cultures. And in any case, all these maps I've been showing you are all available online in this Figshare collection that we uploaded along with the paper. So with using these really cool microrheological techniques, we were able to, for the first time, show that all that was hypothesized about the heterogeneity of viscosity in the microscale was true, that indeed viscosity is heterogeneous. We also show that around the phycosphere is basically viscous too. And in summary, this gives us a really nice quantitative framework to try to understand what is the effect of this viscosity at the microscale. But the big question, at least to me, is what is the ecological significance of this heterogeneity? And this is like opening a can of worms, no? There's so many different processes that can be affected by viscosity, and not just at the microscale, at all scales, no? And some of them have been already explored by Ian Jenkinson and his collaborators, no? For example, it could affect the turbulence cascade, or it could affect the contact rates between different particles. It could affect the aggregation rates and the formation of marine snow, predation, symbiosis and infection of microorganisms with mucus layers. So it is really quite an exciting field, full of possibilities, we believe. Just centering a little bit more on EPS, which is what we are most interested on, there has been described or hypothesized a lot of different roles of EPS in planktonic organisms. And here I list quite a few of them, no? So from attachment to biofilms or to other particles, to metabolic excess waste products, enhanced forward scattering of photons, protection from grazing and trapping of viruses, enhancement of microscale gradients, absorption of nutrients, conservation of exoenzymes, and protection against desiccation, salinity changes, or UV light. No? I think that viscosity is quite relevant to some of these functional roles, and actually could add some more roles to this list. No, so I'm going to center now to what could be the ecological relevance of this EPS on the phycosphere. And to simplify again, I'm just going to consider the two main effects that increasing viscosity may have at these scales for the microorganisms. One is that it will slow down particles moving either actively or passively. No, it will slow down sedimentation, but also it will slow down bacteria swimming. On one hand, and then it will also decrease the diffusion of molecules. So let's go first to the molecular diffusion. No? We had this very simple experiment in which we used a dome chamber that you can see up in this corner, which is a kind of a slide that is used to detect and quantify chemotaxis in organisms. In this case, we use it to look at what was the diffusion of of a solution of fluorescein when we added phycol, which is a polymer that increases viscosity without modifying the Newtonian behavior of the fluid. So in this case, we have the distance from the source of the fluorescein against and the concentration, no, of the relative concentration of fluorescein. No? When there's only fluorescein, we expect, given the geometry of this chamber, we expect a linear gradient. But when we add viscosity, this is not the case anymore. We have a very, very sharp drop in the concentration of fluorescein. And when we look at the dynamics of this, when we look at the time of these chemical gradients without and with viscosity added, we can see that the difference can be quite substantial. No? So it's not only that the gradients are steeper, it's also that the gradients are much longer lived. And this can be very important when you think about how bacteria are going to track these gradients in the ocean. We try to model this in a more controlled manner, no? And so in this case, this is work from Sassi Pasupuleti, who was in our lab using COMSOL. And he modeled what would happen to a spherical cell of 200 micrometers in diameter if we suppose that the wall completely disintegrated. So it's basically a way of modeling a lysing event, no? And here we have, in the right circle, we have the two situations, no? In the top one, what happens if we don't consider viscosity, no? And what you can see is that after 5 minutes, there is a nice gradient of the sugar that he was using. But if we consider at the same time viscosity, which is itself takes quite a long time to diffuse, then again this gradient is quite sharp, and it doesn't go away that fast. So one of the possible implications of this is that the presence of these EPS layer around the phytoplankton can decrease the diffusion of the inorganic molecules that the phytoplankton need to grow, and therefore decrease uptake rates and decrease the growth rate of phytoplankton. No? And this is a really nice modeling work from Bryce Inman that is, I believe, currently accepted in LNO letters, in which he showed that if we consider viscosity decreasing approximately in the manner that we observe in our experiments, that leads to decreases in diffusivity as you approach the cell, which translate into steeper gradients of concentration of the nutrients. And if you do the numbers, that again can translate into lower uptake rates. So that begs the question: why would phytoplankton do something that can decrease their chances of or their growth rate? No, and obviously all the benefits from this EPS must outweigh considerably this effect. Moving on, we also are interested in seeing how viscosity will affect the motility of bacteria. No? Here I have two videos, one of E. coli swimming in a motility buffer. In the left, sorry, in the left one, is a motility buffer without anything. In the right one, we added phycol again to increase the viscosity. No? And so we use these videos and we track the bacteria to understand what was the effect of increasing viscosity on the motility of bacteria. No? And so, for example, here we have the results for low viscosity. This is the histogram of the run speeds, which is typical. It follows along normal distribution, which is typical from bacteria swimming. And then at the right, we have the distribution of the distribution of angles of rotation or tumbling angles. No, don't pay attention to this run speed here because this is wrong. This is the angles of rotation. Okay. And so this is typical from this species, no? That there is a more or less uniform distribution of tumble angles with some bias towards the front, no? But when we look at what happens at the high viscous fluid, we can see, as we were expecting, that the viscosity decreases the speed, the running speed, quite substantially, but also it changes the rotation, the distribution of orientation angles, no? Because it increases the rotational drag of this that these bacteria are experiencing. So it's harder for them to turn. That completely changes the motility pattern of bacteria, no? And putting all this together, we did these experiments with a gradient of different viscosities, and we could see that the swimming viscosity decreased non-linearly with the swimming speed decreased non-linearly with viscosity, and the directional persistence, which is a measurement or a parameter encapsulating this constrainment of the tumble angles, also increased with viscosity. How this translate into their ability to track the gradients? Well, to understand this, what we did is an IBM model, an individual-based model, in which we use the well-known chemotactic pathway models of Escherichia coli. And then we just simulated hundreds and thousands of bacteria swimming using this run and tumble, and we used the observed experimental functional relationships with viscosity. And so we used this model to look at what was the behavior of the bacteria when subjected, or when they found a gradient of viscosity or of nutrients, or both. I have to say this is a quite exaggerated gradient here. I just used a very steep gradient for illustration purposes, obviously, but we did not use this gradient. We actually use, you can see it here in this inlet graph here, you can see the gradients we use. This is the gradient around the phytoplankton cell, and the distance is quite large, 2000 micrometers. And so in blue here, you have the gradient of nutrients when there was no viscosity. Then the gradient of nutrients when there is viscosity, which again decreases the diffusivity of nutrients. And here is the gradient of viscosity. And so what happened here with this IBM model is that, as expected, when when you had a gradient of nutrients, all the bacteria in the domain would tend to aggregate towards the source, the nutrient source, no? And that was relatively quick. Within 5–10 minutes, you could see an effect here. When you added viscosity, if only because the nutrient gradient was steeper, the effect was much larger. So you can see here how dramatic this could be. But even if you don't have nutrients, even if you only had a viscosity gradient, you could see an effect. You could see, and remember, this is 2000 micrometers, but if you look at the first 200 micrometers, you can see increases of up to 50% in the bacterial concentration near the viscous source. And that's what we call viscous trapping, no? So there is a kind of a hydrodynamic effect, no? Just because there is a gradient in velocities, in the velocity of bacteria, induces a change in the distribution of bacteria. Again, this red line is caused by the increase in the steamness of the nutrient gradient, by this viscous trapping, and also because the signal to noise ratio increases as the cell doesn't turn that much, so tumbles are more restricted. To start finishing things, I would if you have to take home any messages here, I guess it would be that viscosity is heterogeneous at the microscale, and we have proved this, and that nutrient hotspots should always coexist with viscosity gradients, because basically they are organic nutrients that are adding some properties to the physical properties to the seawater. At the most basic level, viscosity affects nutrient diffusion and plankton motility, and this can lead to a cascade of implications to the functioning of the whole planktonic system, we believe. And we are just starting to explore all this. So some of the things we are focusing on now is first looking at the viscoelasticity. So these techniques allow us actually to measure the viscoelastic moduli of the fluid. So it just takes it's a little bit more complicated, but we are getting there. So that would add a new layer of complexity to the whole picture, but we think this could be quite exciting. We also want to develop some better understanding of how anisotropic is this system. We also want to see what is the variability in the field. So we are taking samples in the field, and we are using the same techniques to assess what is the variability or the heterogeneity at these scales in real life, no? And finally, we want to replicate what we did with this IBM model in the lab using microfluidics. And for this, Sasi developed this microfluidic device, which is working very well, and we are now in the process of testing with bacteria. This microfluidic device allows us to overlap a nutrient gradient and a viscosity gradient and see what is the behavior of bacteria when exposed to both gradients, no? And finally, we also want to see what is the effect of size of all these molecules and all this. And just as a final thought, we have simplified a lot here, but obviously all these EPS layers are quite complex structures with a lot of pores and chains of very different length, no? And actually, when you look at the viscosity measured with different probes of different sizes of the same, in this case, it's a polysaccharide at different concentrations here, you measure different viscosities. So actually what you are measuring with our beads is not the viscosity per se of the fluid, but it's the viscosity that the probe is kind of experiencing, no? And it depends, this viscosity, what we call this effective viscosity, depends not just on the solvent, the seawater here, but also on the polymer structure in which this bead or this probe is embedded with its porosity, tortuosity, mesh size, no? And also on the size of the probe. So the larger the probe and the more intricate the polymer structure, the larger the viscosity you are measuring. But that doesn't mean that the smallest inorganic molecules will experience such viscosity. So that leads us to believe, or to frame the problem maybe in a different way. No? What if EPS is actually a kind of a tool or for size fractionation of resources? No. So if you think about molecules and bacteria as resources for phytoplankton, having this layer around them of viscous material could allow these cells to recruit and trap chemotactic bacteria that then can form part of the phycosphere. And at the same time, it may not have such a, it will have some effect, but maybe it's not such a strong effect on the uptake rates of small inorganic molecules, particularly if those molecules are mineralized by the bacteria within the phycosphere, no? And then also it has an effect on positive effect on bacteria, because the large molecules will also suffer a decrease in their diffusivity. So exoenzymes released by bacteria, or the organic nutrients with which maybe phytoplankton feeds bacteria, will not diffuse away that fast. So we propose that maybe we could start seeing this EPS as a kind of an engineering tool for phytoplankton that allow them to have more control on the physics and the composition of their immediate environment. And with that, I will leave it, and I would like to thank or maybe blame, I don't know, all my collaborators, or the people, the members, past and present of the Physical Ecology Lab, and collaborators in Scripps, Jules, Jav, Peter Franz, and Pichaya, at the University of Nottingham, Amanda and Tanya, who were instrumental in doing most of the MOT experiments, and also Manlio Tassieri, which gave us a lot of expertise on microrheology, and also the funding of Gordon and Betty Moore Foundation. Thank you very much for your attention, and I will take happily any questions you may have.
Thanks, Oscar. And yeah, I'll open the floor for questions, and you can put them in the chat or just raise your hand, or even just speak up. Marco.
So thanks for the talk. So perhaps a slightly technical question, which actually I might have already asked you in the past, but if that's the case, I didn't register the answer in my brain. So when you do the simulations of the diffusing molecules in the viscosity gradient, there isn't a single way of doing this sort of simulations, because you need to specify where calculate the viscosity, right? So there is, for example, this stuff about Ito versus Stratonovich and blah, blah, blah. So you need, in a sense, to specify how the gradient in diffusivity is going to enter into the dynamics of the molecules.
Yeah.
So for example, if you have just a container with pure water, the molecules will experience a higher drag coefficient when they are very close to the boundary, just because of hydrodynamics, but that does not mean that they accumulate at the boundary. However, if you have a change in temperature across the system, so the system is out of equilibrium, then you do expect to see gradients developing, or you can expect to see gradients developing due to changes in diffusivity. So I was wondering whether you guys have discussed this in any way.
Well, there's two things to your question. One is all these models that are done on with COMSOL, I mean the diffusivity models. So COMSOL takes into consideration the concentration dependence of viscosity. Okay, so viscosity per se is just the manifestation of a certain the presence of polymers that will themselves diffuse, and so the viscosity itself will diffuse in a manner that is dependent on the concentration of these polymers, no? or of viscosity. No? So it's self-dependent in a way. This is all taken into account, and it's included in the COMSOL simulation. About the hydrodynamic, it's just that I'm not sure if you are talking about the two things, or it's all the same thing. But about the hydrodynamic effects, we did take them into consideration when doing, as I mentioned, the microrheology measurements. And they are well described by this semi-empirical equation, the Faxen law. To be honest, I am not sure how could you actually implement this into the.
No, but but I wasn't talking about that. But I think perhaps I'll pick this up another time when we can sit down. Somewhere. Okay, so I'll just
but I appreciate the question, and I would be very grateful if we can discuss these issues.
Cool. Ati.
Yeah, hi. Great talk, Oscar. Thanks very much. It was really interesting. The Stalker group, and also with my student Teresa as well, we've done some work on chemokinesis. So this is when the bacteria swim faster due to a local concentration of chemical. So we completely ignored the viscous effects that you've been considering. So have you considered if the bacteria, for example, could respond to the chemical nature of the EPS and therefore alter their speed?
Yes. In our experimental model, that doesn't happen because I mean, we've seen that in E. coli there is no such chemokinesis. So that is one less thing to take into account. No? But obviously, we are simplifying a lot here. So there are some papers, quite old papers, that talk about viscotaxis, for example, and the ability of bacteria to modify their behavior when they sense a change in viscosity. That would be very interesting to explore. We haven't found that particular behavior in real life, though. But if you have any idea of where to look for, we would be certainly interested in trying that. I must say that when, so this chemokinesis effect does not collides a little bit with what we discovered or we hypothesize about this viscous trapping, no? So we observe this change in velocity. So this decrease in velocity that led to an increase in the aggregation, no? Which is exactly the opposite than that you see with chemokinesis. But obviously, in your case, you find changes in the in the chemotactic behavior, which we don't. But it would be very interesting to maybe to try to reconcile both and see if the fact that they tend to swim faster because there is a steeper gradient somehow contrasts the purely physical effect of viscosity.
Yeah, because I mean some marine bacteria and some soil, marine bacteria and soil bacteria have been shown to be chemokinetic.
Yeah.
And I think there's some evidence for E. coli under certain conditions as well, but by and large, E. coli is not known to be chemokinetic. So,
no, I mean, in our experimental system, at least, we didn't see anything.
yeah.
But yeah, it changes from strain to strain. So,
anyway, I think you've unveiled a really important aspect.
Thank you.
Mimi.
Neat talk. I'm wondering what you've thought about whether this high viscosity halo around a phytoplankton could be anti-predation, that might be one advantage, because predators who sense the presence of prey by smelling them, they would have a much smaller halo. And I wondered if you had explored that at all.
Yeah, this is this is an idea that we have been toying with for quite a long time now. So it's this idea that there is a disassociation between the smell and the food in a way, no? So all these small particles will diffuse away faster than the large ones, no? And so what it means is basically that it may be quite more difficult for predators to find the prey. And I also have to say that this idea has been circulating for a few years now, or for even a few decades. So the fact that in general, the existence of this EPS may hinder predators and may difficult the detection and the approach to the cell. Another question. Yeah.
Oh, I was going to say a flip side is that you're functionally bigger if you've got a viscous layer around you. So if you do get near, say, the filter of a predator, you're much more likely in the feeding current to encounter a capture structure than if you didn't have the viscous.
Yes. Yeah, that's right.
So there's sort of two opposing things going on that might be, it's fun to think about.
Yeah. Yeah, I haven't thought about that, but yeah, that's true too. So there's, as Bryce very well put in his paper, there is pros and cons, no, and the benefits must outweigh the cons, because there are some uptake rates, and what you mentioned also seems like a very important effect.
Yeah, Kelly.
Hi, it was a really nice talk. I really enjoyed it. I was wondering, so there's all the stuff about cross-linking of organic material to form gels, and I was wondering how, oops, hang on, my cell phone rings right while I'm talking. I was wondering what you think, like you said, you're starting to look into viscoelasticity, and I'm interested in how you're doing that, and also if you've thought about what the role of gel formation versus viscosity might be. So if you have gel formation, does that sort of clump the polymers that would be increasing the viscosity and create more heterogeneity or less viscosity, or I'm kind of like going in circles in my head on that, but I was wondering if you've given that thought.
Well, I presented a quite simplified picture, but there is a continuum of behaviors from the purely viscous fluid to the elastic, purely Hookean solid or semi-solid. No, I would expect. I mean, to find the whole thing, the whole range of behaviors in EPS, and we are still working on it. The way where so these microrheology techniques actually allow you to extract, as I mentioned, the viscoelasticity of I mean the range of frequencies in which you can explore this is limited, and maybe we will, if we want to look at what will be the effect of elasticity on bacteria swimming around these areas, maybe we will need to switch to active microrheological techniques. I don't know, but we are working on it. I also should mention, I didn't have time, and so I thank you, your question for this, that the picture we presented about the effects of viscosity on the motility of bacteria was quite narrow. There is a lot of literature that shows very contrasting effects of viscous fluids on the motility of bacteria and other motile microorganisms. So it depends on the shape of the organisms, it depends on the viscoelastic properties of the fluid. And so there are some adaptations that bacteria or motile microorganisms in general can adopt to actually swim faster when they encounter a mucus layer, for example, near corals, just as an example, no? So yeah, the short question is that we are still working on it, but we expect the picture to be extremely complex, and well, we'll deal with it once we have a better picture.
Thanks.
Yeah, so on that note, let's thank Oscar again. Thank you. It was a wonderful
Thank you.
seminar.